This method can help answer key questions in oral cancer research field, such as how to build an appropriate TSCC model in vitro. The main advantage of this technique is that reconstructed TSCC exhibits tumor characteristics that bear a strong resemblance to clinical TSCC histopathology.
We first had the idea for this method when we realized that 2D culture TSCC cells did not properly exhibit the native phenotype of TSCC. Furthermore, we realized the importance of extracellular metrics for cancer survival, evasion, and differentiation.
Visual demonstration of this method is critical, as the preparation of tough extracellular matrix and 3D culture using a mini bioreactor are difficult to learn through words only, and are best seen for clarification.
After isolating the tongues from euthanized mice according to the text protocol, immerse the tissue in 75% ethanol for 3 minutes, then transfer each tongue into a 1.5 milliliter microcentrifuge tube with 1 milliliter of 10 millimolar sterile PBS. To carry out cell lysis, freeze the tongues at -80 degrees Celsius for 1 hour, then thaw the tissue at room temperature for 45 minutes. Repeat the freeze-thaw 2 more times. Next, in a 3.5 cm or 6 cm culture dish containing 75%